Hello students, I am Dr.R.P.Oviya from the Department of Biotechnology, School of Bio and Chemical Engineering, Sathyabama Institute of Science and Technology, Deemed University, and here we will look into what is polymerase chain reaction. So in the previous videos, we looked into what are all the techniques that can be used in genetic engineering. So we can isolate the total RNA, check for the intact and how the RNA is present using agarose gel electrophoresis, and then we can go for polymerase chain reaction where we can amplify the target gene and get more amplicons such that it can be used for cloning purpose. So what is polymerase chain reaction? It is a technique in molecular biology used to amplify a single copy or a few copies of a DNA that can generate thousands of millions of copies such that that particular DNA sequence is only present in the sample. So here the first step is denaturation, where the double-stranded DNA template is now converted to single-stranded by heating. At heating at higher temperature around 95 to 100, the DNA coils will break down and it will form a single-stranded DNA. So now we need a primer. A primer is something that is a short sequence. It is around 15 to 20 base pairs, and this sequence is a complementary sequence of the target gene. So primer can be synthesized in vitro, and this primer is specific for a particular gene. So this is specific for our target gene, and we have to ensure that it gets annealed to the target. So annealing happens at a particular temperature, and then once annealing occurs, the polymerase has to work. The DNA polymerase has to now add the new DNTPs based on the template gene. So for the polymerase to work at higher temperature, it is called Taq polymerase, which is isolated from Thermus aquaticus, a highly thermophilic bacteria. So this polymerase works at very high temperature, and it's a very useful one when it comes to PCR. So here the addition of polymerase will ensure that the polymerase go and sticks to the primer sequence, and then based on the template DNA, the polymerase keeps adding base pairs, that is the DNTPs, nucleotide triphosphates. Each base pair can be added along the sequence as a complementary base. So what are all the components that is present in a PCR? We need the template that is DNA sample, and we need primer. Primer is synthesized by us. We have to design the primer for the target gene, and the primer is being synthesized by us. And we need the nucleotides, DNTPs is added, and we need the Taq polymerase and the buffer, and we need the Mg2+. Mg2+ is essential for the DNA polymerase to work. So these are essential, and in the PCR tube we can perform the reaction in either a 25 microliter reaction or even a 50 microliter reaction. So this is a thermal cycler where the PCR reaction is being set up. So the first step is denaturation, that is obtaining single stranded DNA, and then primer is added, that is annealing. During annealing, the annealing temperature varies from 50 to 60, that depends upon the template, that depends upon the primer that is being added. So this annealing temperature has to be standardized for each of the target gene. And then we need extension. Extension time is at 72 degrees Celsius, and extension depends upon the target base pair. Suppose our target is around 1000 base pairs, we need a minute. For 1000 base pairs, 1 minute extension is required, whereas above 1000 base pairs, the extension time has to be increased. Then the same cycle can be repeated for 25 to 30 cycles in order to obtain more amplicons. So here we can see that the original double-stranded DNA is there. Now it is converted to single-stranded DNA. From single-stranded DNA, the primer is added. The primer binds and the DNA is synthesized, and each cycle, the cycle 2 is the synthesis time, and each cycle can generate exponentially. So that this temperature is set up by us, and the reaction must be standardized, the temperature and the time must be standardized for each particular target gene. So this is how the pattern looks like. Here we can see from the starting, we can see the denaturation takes place at 100 degrees, around 95 degrees, it has to get denatured in a minute, and then suddenly the temperature drops down to annealing. Annealing temperature is around 50 to 60, and then once the annealing is done for a minute or so, then it goes to extension. In the extension, the polymerase keeps adding the DNTPs so that the complementary sequence is added, and then the next cycle proceeds. This cycle is 25 in case of polymerase chain reaction, whereas for real time the cycles can be even more. It even can be done for 40 cycles. So once the cycle is done, once the PCR reaction gets over, the PCR product can be visualized using agarose gel electrophoresis. In the previous video, we showed how the agarose gel electrophoresis can be run, and using ETBR as an indicator dye, we can check for the target gene expression. Here we can see how the target band is present. Suppose we get a multiple band that shows that our PCR reaction is not standard or optimized. So to get an optimized reaction, we have to set up the particular annealing temperature. Post standardization of the annealing temperature, we can get a single dark band which shows that our PCR is right. So once we get this band, we can go for cloning. This target amplicon can be used for cloning purpose. I hope you understood how we get the target gene amplified, and post this how in our previous video I showed about how the cloning can be done can also be understood. Thank you.